Okay. We are going to start chapter 17 today and get halfway through-ish, is my guess. So the goal is to finish 17 on Friday, and Monday we might actually start 18. We'll see. No quiz on Friday. Your homework is going to be due on Monday for chapter 17 and 18. That is the last homework assignment. I debated putting it off, but you don't want to do the Monday after break. We're just gonna get it done. So 17 and 18 are gonna be together. It's gonna be posted later today. I'm almost done with it. Right now. I was talking to someone this week and looking through the book and talking about different ways to study, and I was like, oh, look, there's questions at the end of the chapter, and then I was like, oh, look. So there's actually like a little website there that has a review questions on it that you can go to. And it's got a lot of review questions that are multiple choice. And then there's also a separate one that's like vocabulary review that are flashcards where you can put the definition up and then try to guess what the word is and flip it over. They're all made, or they're all from the book, and they're all at the end of the chapter. So in that chapter summary part, they're all there. You do have to type in the website. The old version had a QR code, but the newer version does not have a QR code, but they're listed there. So that's another resource that you can use for studying. Our last test is probably going to be Wednesday after we get back from break. Thanksgiving is really late this year. It's inconvenient. I didn't schedule Thanksgiving. But Wednesday after break. Then Friday we'll spend reviewing, and then our final Monday at 8 a.m. of finals week. So there's a lot coming up. And if you have questions about the schedule, let me know at any point. So your review question from Monday, because their day is Wednesday, is what is the central dogma of biology? You can all tell me it because you think about it all the time. The central dogma of biology said DNA makes RNA, RNA makes protein. So we spent some time in chapter 16. It's really, if we write it all out, I have very few classes left to master the not advancing the screen accidentally. So DNA also makes DNA. That's the process of DNA replication. That's what we talked about in chapter 16. Chapter 17 is covering the second part, the DNA making RNA and the RNA making protein, which is a two-step process. And that's what we're going to be talking about today. Starting to talk about today. So this is what we call gene expression. So taking DNA and making proteins out of it is what we call gene expression. So we're taking the instructions on the DNA and we're turning it into a product. This is also what you're talking about in lab this week. Those of you who had it yesterday already did this, but it's the idea of the genotype, meaning the DNA, determining the phenotype, meaning the proteins that are present. So this week in lab, you're taking little fruit flies and squashing their heads and then running chromatography to see what pigments they have, what proteins they have, based on what genes they have. So the genotype is determining the phenotype. And that's gene expression. That's the central dogma of biology. That's the whole thing. So we have two pictures here showing you some different animals that have a mutation in their genes. They have a gene mutation that means that their body doesn't produce the pigments, the protein pigments that would give them coloration. So you've got albino donkeys on the left and an albino raccoon on the right. It's miraculous that an albino raccoon survived to reproduce because it looks noticeably different and would be easier to find if you were hunting raccoons. But then I was thinking, like, who hunts raccoons? I mean, people hunt raccoons, but like, what is the predator for a raccoon? Coyotes? Wolves? I don't know. I don't know if there's any natural predators for raccoons in our area. Coyotes, maybe. But that one would definitely stick out. The albino donkey population, they talked about it in the introduction, is from an island off the coast of Italy. I think that just there was one albino donkey that survived and reproduced, and then all of a sudden now there's a large population of albino donkeys. But that's again the genotype determining the phenotype. So this is our overview of this process. And we're going to talk about it from both a prokaryotic standpoint and a eukaryotic standpoint. Focusing a little bit more on the eukaryotic cell. So on the left, we have a prokaryotic cell. And in this case, when we're talking about prokaryotic, we're talking about bacteria, not archaea.
archaea, which we will talk about next semester. So there are some differences between prokaryotic and eukaryotic cells in the ways that they approach this process. But there are also similarities. So the first part is showing us we have DNA. In the prokaryotic cell, where's the DNA located? Does it have a home? It doesn't have a nuclear envelope surrounding it. It's in one region called the nucleoid region, but it doesn't have a home. So it's a little bit different already from the start, just structurally, because there's no nucleus for the DNA to be housed in. So when we take DNA and we turn it into RNA, so this is our DNA molecule, and this is our RNA. We're specifically talking first really about making mRNA. That process, we're staying in the same language. So this is called transcription. We can't see that. So when we talk about transcription, whether we're talking about it in a prokaryotic cell or a eukaryotic cell, we're talking about taking DNA and changing it to RNA. And again, we're staying in the language of nucleic acids. So we're not changing languages, we're changing the form of the language. There are people who act as scribes for doctors. So they listen to what a doctor says and then they write it down. So they're still dealing with the same language, English or whatever other language they're talking in, but they're a different form of language, oral versus written. So it's transcription because you're not changing the message, the way the message is expressed. It's still in the same language. You're just changing the form of the expression. So in this case, we're staying in the language of nucleic acids. We're taking DNA and we're making RNA. From there, the RNA is changed to the language of proteins. And this is translation because we're using, instead of the language of nucleic acids, we're now using the language of what kind of acid? If it's a protein. What? What kind of acids are in proteins? Amino acids. So yeah, it's translating nucleic acid language into, I'm just going to write that here too. So translation, we're switching languages. We're going from RNA, to a polypeptide or a protein. So we're going from nucleic acids to amino acids. And you'll notice there's an extra step here in the eukaryotic cell. And that eukaryotic cell, when we make the mRNA, oh, that doesn't show up. So when you have the DNA and we're changing it to mRNA, there's actually an extra step, which we'll talk about in a little more detail as we talk about all of this in a little more detail. That RNA that is initially made is processed from what we call a pre-mRNA. So this is really pre-mRNA to a mature mRNA. That doesn't happen in bacterial cells, but that does happen in eukaryotic cells. It enables the mRNA to be able to leave the nucleus and be more stable when it's out in the cytoplasm. So we're carrying this genetic message from the nucleus out to the ribosome so the ribosomes can do their job in synthesizing proteins. A little bit of background on how we got to this, how we figured this all out, because we like to talk about experiments. We've talked about like a million experiments already in figuring out the structure of DNA and DNA replication. Some work was done in the early 1900s. With the guy's last name is Gerard, and he basically developed what we called the one gene, one enzyme hypothesis. Which was that every gene coded for an enzyme, and one gene coded for one enzyme, a different gene coded for a different enzyme, and each gene was specific for only one enzyme. That was the working model for about 60 years. Well, no, more about 30 years, sorry. The 60s is when we figured out some other stuff. In the 1930s, a scientist named, whose last name was Beadle, was working with fruit fly eyes with a guy named, I don't know how to pronounce this. I'm not Italian, Frusi. So they were working on fruit flies and fruit fly eye color. So expanding kind of on Morgan's idea that fruit fly eye color is inherited and that there are different mutations. So working with fruit fly eyes, they determined that there was a whole long pathway and that at each step, there was an enzyme that was necessary to make the pathway go forward. This is exactly what you're looking at in lab. So you're looking at 6 different colors of fruit fly eyes and seeing what happens if there's a mutation in the gene. How does that fruit fly's eye look different? And so you have a nice complicated metabolic pathway that you're looking at and figuring out, okay, if the mutation happens here, then this is what happens. If the mutation happens here, this is what happens. So you're doing the same scientific work or similar scientific work to what they were doing. But this was the idea that there was an enzyme for every step of a metabolic pathway. That wasn't really Beadle's most famous work. He went on to do further research with the last guy whose last name was Tatum. And they did work, instead of working with a diploid organism, they used a bread mold. And it's a haploid cell. What do we mean by haploid? It has, yeah, only one copy. It has half the number of chromosomes, so it only has one copy for each cell, for each gene. So it has one copy of each gene, and they developed mutants that were nutritional mutants. So regular old bread molds can grow on what's called minimal media. It's just media that has minimal nutrients in it. But they mutated different genes by exposing them to X-rays. They created different kinds of bread molds that needed extra nutrients. So some of them were deficient for certain amino acids. And so they determined that all of these different mutants had not just one gene, one enzyme, but it was really that each gene coded for a polypeptide. That was their working hypothesis, or what came from out of their research with the nutritional mutants. Why are we saying polypeptide instead of protein? What is a polypeptide? Way back chapter 5, 6. Polypeptide, what, sorry? Somebody said something. A polypeptide is the chain of monomers. It's the polymer of all the amino acids. So remember, the primary structure of proteins is that order of amino acids. And that's really what the gene is coding for. The gene codes for the order of amino acids, so the primary structure, the polypeptide, it's the chemical interactions that occur between amino acids and between different polypeptide subunits that determine the other layers of structure. So the gene is really coding for the order of the amino acids. So when we say DNA makes RNA, RNA makes protein, we're really saying DNA makes RNA, RNA makes polypeptides, but we just kind of simplify it by saying proteins. So this one gene, one polypeptide hypothesis was kind of the working model. It's still kind of our working model. It's not perfect. Some genes actually code for RNA that has functions in the cell, and we're going to talk about alternative splicing in the next chapter, in chapter 18, because you can take an mRNA molecule and you can cut it up different ways and end up making different polypeptides out of the same pre-RNA molecule. But that's the basic idea. is that a gene codes for a polypeptide, one gene codes for one polypeptide. You have to have a different gene for a different polypeptide. So differences between eukaryotic and prokaryotic transcription translation. Main difference is there's no nucleus in the bacterial cell. This means that both processes can happen at the same time. So in a eukaryotic cell, the mRNA is made and the nucleus has to leave before it can be translated. In the prokaryotic cell, it can all happen at one time because there's nothing there. There's no barrier. So transcription can be happening and translation can be happening immediately afterwards. So these two processes in a prokaryotic cell can happen simultaneously. The other main difference is this step of RNA processing. That in eukaryotic cells, RNA has to be further processed before it can exit the nucleus and be used. So we talk about central dogma, DNA makes RNA, RNA makes protein. There is actually, this is just a side note, a reverse process. RNA can actually go backwards to DNA, and there's an enzyme that makes that happen. Called reverse transcriptase. Why does that name make sense? Reverse transcriptase. Why does that name make sense?
Reversing the transcription.
Yeah, transcription takes DNA to make an RNA. If you're going to reverse it, we're going to take RNA to make DNA. And this is really important with viruses, which we're not talking about right now. We'll talk about that next semester. But I just thought that I think it's interesting that it can go both ways. Couple reminders before we get into all of this. There's some differences between DNA and RNA, which we've already talked about. What are the three main differences, I would say, between DNA and RNA? So between DNA and RNA, DNA, I think you mean helix. DNA is a double helix. RNA is a single helix. So DNA has two sugar phosphate backbones. RNA only has one sugar phosphate backbone with bases sticking out. What else? Yes. RNA has uracil, but DNA has thymine. You know, there's the no I in team. I always think of there is no U in DNA. I don't know why they don't make shirts like that. One other major difference. The sugar that's present. So they both have a sugar phosphate backbone. In DNA, the sugar is deoxyribose. And in RNA, the sugar is ribose. The main one that's going to be important for us as we talk about this is the thymine uracil thing. So in DNA, A bonds with T, but in RNA, A bonds with U. So when we talk about complementary bases, you have to think about when you're dealing with an A, you have to think about if you're talking about DNA or RNA, because it's a different bond. Okay, one more overview. And then we'll get into the details of transcription. So what are we starting with at the top? This is our DNA, which has two sides. Sam said to me as I left, you always take that with you. I'm like, we're still talking about it. Okay, so same kind of thing is going to have to happen. The DNA is going to be exposed, unwound, the hydrogen bonds between the bases are broken, but only one strand acts as a template strand. So only one strand is going to be transcribed. The other strand is called the non-coding strand. So the top strand is in this case, this is our template strand. This is our non-coding strand. So in a long strand of DNA, our DNA is linear. There's a lot of different genes, and those genes are, sometimes you read the top strand and sometimes you're reading the bottom strand. So at different sections of the DNA, the strands might be transcribed. The origin sequence. So there's a specific sequence that the RNA is looking for, the RNA polymerase. That origin sequence determines which strand is the one that's transcribed. And again, in different parts of the DNA, it could be a different side. The coding strand or the non-coding strand is going to have the same sequence of bases as the mRNA, except for one difference. T will not be present. The mRNA has a U. So we're going to code this strand. So this and this are going to be the same. The non-coding strand and the mRNA are going to have the same order of bases. So again, we're doing transcription. Which means we're taking nucleic acids and changing to the other form of nucleic acids. So if we take this strand of DNA apart, and we're going to read the top part, pretend you can't see this. A is going to pair with what in mRNA? A is going to pair with U. C is going to pair with G. C is going to pair with G. A is going to pair with, not all of you at one time. A is going to pair with U. A is going to pair with U. C is going to pair with, C is going to pair with, G is going to pair with, A is going to pair with, G is going to pair with, and T is going to pair with. So anytime we have an A in DNA, we get a U in mRNA. But if we have an T in DNA, we still get an A in RNA. So we change this language to mRNA. M stands for messenger. I don't remember if I said that. Which makes sense. It's the messenger. It's taking the message of the DNA from the nucleus out to the ribosomes. mRNA is going to be complementary to the template strand. So it's not the same. It's not taking the exact message. It's taking the opposite of the message. And this is going to happen just like what it did in DNA replication. It's anti-parallel synthesis. So the enzyme that's going to bring in those bases can only add it to the 3' end, just like it could only add it to the 3' end. DNA polymerase 3 can only add to the 3' end. So we're going to add nucleotides to the 3' end of the template strand of DNA. The mRNA is, yes, of the mRNA. Okay, the second process, then changing that language, is translation. So this is taking nucleic acids to amino acids. And it has to be read in chunks, and those chunks are called codons. So it's a triplet codon because it's read in sections of 3. So each set of 3 base pairs, each set of 3 bases is going to code for 1 amino acid. 3 base pairs, 1 amino acid. 3 base pairs, 1 amino acid. So we have 4 different codons here. They're coding for 4 different amino acids. The primary transcript, this is called the primary transcript, is going to be read in a 5' to 3' direction. So it only can be read one way. It can't be read backwards, which makes sense because if you read it backwards, it would be the opposite, a different message. It wouldn't be the same message. This triplet codon is important. Because how many amino acids are there? Anyone remember? 20. So there's 20 amino acids. So we can't have. We have to code for all 20 amino acids. If we just read one base pair at a time, there's only 4 different base pairs. So we'd only be able to code for 4 amino acids. If you read 2 base pairs at a time, there's only enough combinations for 8, I think, if I remember right. Yes, no, yes, 8. There's only enough for 8 amino acids. So 2 base pairs at a time wasn't enough. So it has to be 3 base pairs at a time. But when we do 3 base pairs at a time, we have 64 possibilities. And those 64 combinations are coding for 20 different amino acids. So 64 combinations, 20 amino acids means that we have more than one codon for amino acid. For some amino acids. Not all amino acids get as many as the other ones. It's not evenly divided. So you'll see here like UUA, UUG, CUU, CUC, CUA, CUG, all 6 of those code for leucine. There is one that is only got 1. In addition to the 20 amino acids, 61 of these codons code for amino acids only. 3 of them are what we call stop codons. So that's what's going to stop the process of transcription or stop the process of translation. UAA, UAG, and UGA are all stop codons. One of these amino acid codons is a special one. The codon AUG codes for the amino acid methionine, but it also is the start codon. So all translation starts with AUG. So every mRNA has to have an AUG at the beginning to start the translation process. So we refer to this as the genetic code is redundant, meaning there are multiple amino acids for multiple codons for the same amino acids. We also use the term degenerate. The genetic code is degenerate for the same reason. More than one amino acid can code for the same, more than one codon can code for the same amino acid. You'll notice that in a lot of these, the difference comes, in a lot of the similarities, the difference comes in the 3rd codon. So for proline, for instance, CC, the beginning 2 codon CC, all code for proline, but the end one doesn't matter. It can be U, C, A, or G. They all code for the same one. And we'll talk about why that's actually helpful. You can think of this as the idea of like synonyms. They're different codons that mean the same thing. The other thing that's really important about this is the idea of reading frame. You have to read it in sets of 3, and you have to start with the right one. If you're trying to read a sentence in a book and you skip the first letter of the first word and you only read in sets of 3, it's not going to make any sense. You're not going to get anything out of it. So the reading frame is important to get the right message across. So when the mRNA is being read, it has to be read in sets of 3 and has to be read in the right sets of 3, in the right order to be able to get the right one. This whole thing was figured out in the 1960s. So this table was developed through research at the NIH. A guy named Nuremberg figured out the first codon, and from that, throughout the rest of the 60s, they figured out all the rest of the codons. Again, you worked with this some in lecture. You'll do some with this on your homework as well. I think it's fun. The amino acids also get 1 letter abbreviation, so there's 20. So I like to try to write words with that. You can write notes in code. This is what nerds do when they're writing notes. They try to write notes in genetic code to other people, but you have to make some substitutions because you only have 20 letters to work with. Okay. I think we've overviewed enough since it's been half an hour. We're never. So So let's talk about the process of transcription. And when we talk about transcription and we talk about translation, we're going to talk about 3 stages for both of them. Both of them have the same 3 stages, which are initiation, elongation, termination. You have to start the process, you have to make the thing longer, and then you have to stop the process. So initiation, elongation, and termination. This is section 17.2 in the book. So you can see here, we've got our transcription unit. So this is the DNA. We have a specific region of the DNA that's called the promoter. That's going to promote the binding of the enzyme that we need here. And the enzyme that we're talking about now is RNA polymerase. So in DNA replication, we talk about DNA polymerase, and transcription, we talk about RNA polymerase. There's multiple forms of RNA polymerase. We're really only focusing on RNA polymerase 2. Prokaryotes only have 1 RNA polymerase. There's 3 RNA polymerases in eukaryotes, but we're worrying only about polymerase 2. So this RNA polymerase, once it has bound to the template strand, it's going to unwind it, just like helicase did in DNA replication. It's going to break the hydrogen bonds and open it up so that the template strand is available for the complementary mRNA bases to come in. And you can see here we have the beginning of the RNA transcript forming. Then the polymerase is moving down. It's elongating that amino acid, not the amino acid, the nucleic acid chain. And it's going to keep going downstream. That's the direction it goes, synthesizing in a 5' to 3' direction until it hits this terminal code, the stop codon. And that's going to make the RNA polymerase detach. And then we have a completed mRNA transcript. A little more detail on these steps. So looking at transcription, when we talk about initiation, specifically in eukaryotes. I don't want this here. So we'll look at the structure of a eukaryotic promoter. So this is showing us our promoter region. And there's a very specific sequence of bases that's found in the promoter region for all of transcription. And this is called the Tata box. That's something that I remember very clearly learning about the Tata box in college and then not remembering really what it was, but just remembering Tata box. The Tata box is part of the promoter region. So this is our non-template strand on the top, or the non-coding strand. This is our template strand on the bottom. Because again, it can only happen in one direction. So the first thing that's going to happen after the promoter region, we can identify the promoter region, is you're going to get a series of proteins that come in. These proteins are called transcription factors. So the transcription factors are going to bind to the Tata box area of the promoter. These proteins are necessary. They're the necessary support that RNA polymerase needs to be able to bind there too. So once those initial transcription factors bind, that enables RNA polymerase to come in and bind. They help them to bind in the correct orientation. So they're there to help get the RNA polymerase in the right position to bind, to go in the correct direction, to make sure that transcription happens in the right way. So the RNA polymerase is going to bind to the promoter with some other transcription factors. So a couple more proteins come in to help create what we call the transcription machinery. That's not the right word. Initiation complex. That's the word I'm looking for. So this is the RNA polymerase 2 that's binding. Here's additional transcription factors, which we call TFs. And then this is the beginning of the RNA transcript. So initiation happens, transcription factors, RNA polymerase, more transcription factors, start to unwind the DNA and get those first couple of bases in there. Brandon, did you have a question? I said that transcription factors help the RNA polymerase bind in the right way so that it's oriented in the right, it's in the right place and the right orientation. So it's going in the right way. And then more transcription factors help to keep it stabilized and ready to go. So after we initiate, the second phase is elongation. So elongation elongation is just when the mRNA transcript keeps being formed, so we're getting more bases added. As the RNA polymerase moves down the template strand of the DNA. So about this region that's open here is about 10 to 20 nucleotides long, are open and ready for the RNA to bind, to come in and the RNA bases to come in and bind. So it's unwound for about 10 to 20 base pairs at a time. As the RNA polymerase moves down, this is going to rewind. The double helix is going to reform. Now, last time I asked you to come up with a number of how fast you think that happened, and you all grossly overestimated how fast your cells were. Okay, so we said DNA replication happens at a rate of about 50 nucleotides per second. How fast do you think transcription happens per second? How much? Very close. A little bit higher.
31.
No, 35 is close and a little bit higher. It's about 40. Okay, so this is happening at a rate of 40 nucleotides per second. So DNA replication is going on the nucleus, 50 nucleotides a second. This transcription process is happening at 40 nucleotides a second. It can happen simultaneously. In different locations on the same DNA. So RNA polymerase can be bound to the promoter here and start moving its way down. And then another RNA polymerase can come in and bind and start moving down so that multiple RNA polymerases are transcribing at the same time. So you can get multiple transcripts forming at the same time. So it's fast, not just 40 nucleotides a second fast, but multiple of that happening at the same time. This continues until we get to the termination point. Have you all seen the Terminator? I show my age with my movie references. I don't know if I've actually seen it. It came out in 94. 84, 94. It came out in the 1900s, in the 80s or the 90s. I don't remember. So termination happens when the RNA polymerase hits one of those stop codons. Remember, there's 3 stop codons. So when it hits one of those stop codons, that's going to stop the termination process. So we're looking for a specific sequence in the DNA which codes for the stop codon. So that DNA sequence is going to pull in the right bases to make the stop codon, and that just causes that makes the RNA detach. The RNA polymerase will detach from the template strand in prokaryotic cells. This is the simplified one. In eukaryotic cells, it's a little more complicated because our cells are just a little more complicated. When the RNA polymerase hits the specific sequence of DNA, that sequence of DNA is called the polyadenylation signal. Sorry, polyadenylation sequence, which codes for the polyadenylation signal, which is AAUAAA. Notice it has a lot of A's in it. Poly meaning many, adenylation, many adenines. Okay, so that specific sequence of DNA just causes this mRNA transcript to be cut off. It actually cuts it off. So RNA polymerase kind of keeps going down the DNA, but there are specific enzymes that are going to cut it at that sequence. Eventually, the rest of the stuff that's made that isn't really needed is broken down too. But the RNA transcript, when it hits this polyadenylation signal, gets cleaved, meaning cut off. And now what we have in this case is a complete pre-RNA molecule. I guess I should say transcript. That's more specific. So we've made a complete mRNA, pre-mRNA transcript, but it's still called pre-mRNA. This isn't the final product. And we're going to get through the final product because we'll talk about how the mRNA is processed and then we'll be done with transcription, which is good. So this is section 17.3 in the book, which talks about how the RNA is processed. So at this point, where is the mRNA? Where in the cell is this located? Where did this whole process happen? The nucleus, because the DNA is in the nucleus. Okay, DNA can't leave the nucleus, so this has to happen in the nucleus. So mRNA processing is also happening in the nucleus because this is what's getting the mRNA ready to be able to leave the nucleus. There's two different ways we're going to talk about processing. One is altering the ends of the mRNA. Oops. Shouldn't talk while I'm writing. And the second way is by splicing the mRNA or the pre-mRNA. So when we talk about altering the ends, there's two ways we can do that because there's two different ends. So one end is the 5' end. And what do you see here that we've added to this 5' end? What base are we adding? What letters does that say? G, yes. We're adding a G, a guanine, a GTP, because it's got 3 phosphates bound to it. This is what we call the 5' cap. So the 5' cap, we add a modified guanine to the end. And this actually happens as it's being made. This happens after between 20 and 40 mRNAs bases are added, the 5' cap is put on. At the other end, being the 3' end, what do you see here? lots of A's. Okay, we're adding a tail. So this is the cap and this is the tail, and it's called a poly A tail because it's made up of lots of A's. $50 to $200 AAs, 250, sorry. These are added after that polyadenylation sequence. The A, U, A. And then we stick on 50 to 250 more A's. Why do we alter the ends? Your cells are efficient. They're not doing things for no reason. They're not using resources for no reason. Doing these things helps in 3 ways. So altering the ends helps. It helps to export the mRNA, which is now mature. So it helps get it out of the nucleus. It helps protect it as it is. It helps it be exported. Sorry. The second thing is once it's out there, it helps to protect it. So it helps protect it in the cytoplasm from enzymes that might try to break it down. So your cell did the work to make the correct mRNA transcript and it wants to protect it. So both the 5' cap and the 3' poly A tail can help protect it from those enzymes in the cytoplasm by marking it as this is important, don't mess this up. And then it also helps with binding for the ribosomes. So oh no, sorry, it's not the 5'. The ribosomes, once the mRNA is out there, the ribosomes have to bind to it to be able to read it in those codons, those sets of 3. And so that 5' cap helps the RNA, the ribosome bind to the RNA. So it helps it be able to be exported, it helps protect it in the cytoplasm, and it helps the ribosomes bind. So on here, this is the 5' cap. There's also some what we call UTRs. So there's a 5' UTR and a 3' UTR. A UTR is an untranslated region. So untranslated doesn't mean untranscribed. So it's been transcribed still from the DNA, but that's a region of the mRNA that's not going to be translated into the polypeptide. So it's not going to be translated from nucleic acids to amino acids. So it doesn't function in coding for the protein, but they also do help with binding. With binding for ribosomes. And then the second way that the RNA is processed is by splicing. So sections of the pre-mRNA are actually cut out. And then the other regions are put back together, and we'll talk about that better on Friday. Because I want to make sure we talk about it well instead of trying to fit it in 10 seconds. So we'll finish talking about RNA splicing, and then we'll get through translation on Friday.